Okay, so Kévin is from KTH. He will present, he will give a talk on Gaussian process modeling of bioprocesses. Kévin, the floor is yours. We don't hear you, Kévin. I don't hear you, Kévin. The microphone seems to be switched on, but I don't hear you.
On te voit, mais on ne t'entend pas.
Maybe there's a problem with your mic.
Can you hear me now?
Yes.
Yes, okay. Up.
Great.
Sorry. Okay, let's try again. Share screen and so on. Okay. Okay, so sorry. So I'm going to start again. So thank you, Rashid, for the kind introduction. So today I'm going to talk about an application of identification for bioprocesses. So it's a common work with people in KTH from the automatic control and the biology domain. And so basically I'm going to start my talk with I know COVID. You have heard a lot about COVID, and here I'm going to talk about it because maybe you have heard about this, but recently the European Commission authorized 2 new treatments against COVID, which are based on monoclonal antibodies, which are proteins, which will help to inhibit the viral infection. So basically here in this illustration, you have a human cell and a virus of COVID, which wants to enter in the cell in order to duplicate itself. And in order to do this, what this virus should do is to bind from the receptors of the human cell. So the receptor in red of the virus should bind on the human cell. But now, if you add monoclonal antibodies as a treatment, what is happening is that these monoclonal antibodies in green bind themselves on the virus. And so then now the virus is not able anymore to bind on the human cell. And that's how basically the monoclonal antibody treatment works. So how to produce this monoclonal antibody? So basically we use cells from Chinese hamster, more precisely from the ovary. And so what you do in order to produce this protein of interest is that you give some food to the cell. So it will be sugar, amino acids, and also some oxygen. And then the cell is going to produce some MAB, some monoclonal antibodies, and other products also, so lactates and so on. And basically this, what we do is that we take these cells, we put them in bioreactors, and then we try to cultivate these cells in this large-scale bioreactor. So here you have an illustration of one bioreactor of a capacity of 10,000 liters. So it's really, really large-scale application of the cultivation of CHO cells. And a scheme of a bioreactor could be described as this. I'm going to be brief here. So basically here you have the medium where you cultivate your cells. You include some, you bring some air, some oxygen for the cells. You also control the temperature. And what you are going to do is that you are going to bring a medium, which is called the feed medium, so the food, basically, to the cell. And you are also going to take some medium out in order to take the product of interest, which is the monoclonal antibodies. And the living cells, which are in this effluent, we put them back in the bioreactor. So we have kind of a loop system here. Okay. So the problem with MAB is that the production is really, really expensive. And so are the treatments derived from these proteins. As an example, one of both treatments that have been approved by the EU Commission 2 weeks ago is estimated at 1,700 euros per dose, which is kind of expensive. So the goal would be to reduce the price by maximizing the yield of MAB produced by the cells. So that's the idea here, is that in a bioreactor, we want to increase the productivity of the MAB. Question, of course, since we want to do an optimization, the problem is which variable should we change in order to perform the maximization? And here, what we are going to consider here is the feed medium. We are going to try to find the optimal concentration of the chemical compounds inside the feed medium, so the sugar, the amino acids, and so on, such that the cells will produce and at a maximum rate the MAB. Okay, so we know what we need to optimize, but how should we optimize? Well, we could consider first to do an experimental optimization. So we try one feed medium, we measure the MAB, and if it's not perfect, we try another one and we try to do it like this. But this is too long and very expensive. For instance, if you want to cultivate some cells and have some data, you need to wait 2 weeks. And also the price that you have to pay is kind of expensive. So the solution that here we consider in the project is to do a model-based optimization. So since we want to do a model-based optimization, we need a model. And here what we want to model is basically the relation between the concentration of the chemical compounds in the feed medium and the concentration of the metabolites in the bioreactor, including the ones inside the cells. And here what we want to model is the kinetics of the chemical reaction that are happening inside the bioreactor. Sorry. Just for the vocabulary, I'm saying chemical compounds, but I'm also going to use the word metabolites, just to simplify. So this model, of course, we should satisfy some first principle because we want a model which has some biological meaning. And here, the first principle we are going to consider is the mass balance equation. So it's kind of very simple, is that the evolution of the concentration of the metabolites inside the bioreactor is equal to what the cells produce or consume. So this, the sign of this can change. If you have a consumption, it will be negative. If you have a production, it will be positive. Plus what the feed medium is going to bring to your bioreactor, minus what the effluent is going to take out from your bioreactor. So if we put some mathematics behind this by denoting by x the concentration vector, so the vector of the concentration of each metabolite in the bioreactor, then we obtain an ODE where Q is what we call the uptake secretion rate. So it's the quantity of metabolites consumed or produced by the cells during 1 day. Here you have the flow rate of the feed medium. Here the flow rate of the effluent. X feed is the concentration of the metabolites in the feed medium. And it's this variable that we want to optimize in order to increase the uptake secretion rate of MAB, the monoclonal antibody. And yeah, that's it. So that's basically the equation that we are going to work on. And everything seems to be known here, but the only thing that we need to model here is this uptake secretion rate, which depends on the concentration inside the bioreactor. So in order to do this modeling, I will briefly introduce 3 ingredients we need in order to build this model. We need to model the kinetics of the chemical reaction. So for that, I'm going to introduce 3 things, so which are the metabolic network, which basically gives the connections between the metabolites, so the reactions basically. Then we will introduce the stoichiometric coefficients of the reactions, and we will have what we call a stoichiometric matrix. And at the end, we will also introduce the rate, so the speed of the chemical reactions. So that's kind of abstract. Let's consider some illustration in order to illustrate these 3 ingredients. So let's consider that we have a cell which has this form with 5 metabolites. So we have 2 metabolites inside, 3 metabolites outside. And so basically a metabolic network is just drawing the connections between the metabolites, like this with arrows. And so basically reaction 1 is like S1 will be consumed, sorry, and we will produce C1 from reaction 1. Okay. Of course, what I showed you here is really, really simple. In real life, the network that we consider looks like more like this. So everything that is in black is a metabolite, and then all the color arrows you can see is reaction. So that's more kind of a complex modeling here. Then after having considered the reaction, we have to consider the stoichiometry coefficient in the reactions. So if I consider back my very simple example, so here are the stoichiometric coefficient that we have decided in the model. And then I can display this information in a matrix by putting the reaction in columns and the metabolites in rows. So that then if we look at the first reaction, we see that S1 is consumed with a stoichiometric coefficient of 1. So we will put -1 in this matrix, while C1 is produced twice for one S1. So we will put 2 in this stoichiometric matrix. So then when we have this stoichiometric matrix, the quantity that we want to model can be put into this form. And Y is basically the vector of the speed of each reaction. In my example here, Y will have a dimension of 3, and I will have the speed of the reaction. So now since this is quite known from biology, because a lot of studies have been done in this, what we want to model now is the rates. And for the rates, this is where the identification comes, because what we are going to do is that we are going to consider parametric expression for the rate vector. So my each entry of this vector will be modeled with a parametric expression, depending on the concentration inside the bioreactor. And here for biochemical reactions, the type of kinetics we consider, so the parametric expression we consider, are called monad functions. So basically your rate, so the eth entry of Y is expressed as a product of functions, which depends on the concentration. But here, the thing is that it will depend only on one concentration. And here you have some parameter which parameterize the function, this function. And the modulation function can take 4 possible forms, either an activation, so this form, either an inhibition, either double component, or a neutral effect. So just as you can see, is that the double component function can model also the 3 other ones here by putting theta ij or mu ij equal to 0. So basically the parameter vector here contains theta ij and mu ij. And so what we want to estimate is these parameters here. Okay, so in order to do some illustration of what are activation, inhibition, double component, and neutral effect. So here you have some plots. So basically an activation function is like this, is that if your metabolite is more and more present, then your reaction is going faster. While if for an inhibition, if you have more and more metabolites, then your reaction will go slower. So as an example, if you have too much lactate in the bioreactor, the cells are going to die. So we will have an inhibition effect of the lactate on the growth rate of the cell. Double component is activation and inhibition effect in the same time, that you should not put too little or too much of metabolites in order to increase the reaction. And the neutral effect simply means that your metabolite does not have an effect on the rates. Okay. So at the end, the final mass balance equation we have with the kinetics is this one. And what we want to estimate is this parameter vector eta, which is the concatenation of all parameter vector eta ij. Okay. And our goal is to identify this parameter. But the nice thing here, and this is what is done in bioprocesses, is that everything in this equation is known or measured. So what you can do is that you can directly estimate the data of Y, so the vector of the rates of the chemical reactions. And so what you have at the end is this data set, this quantity that we want to model. You have the data of the concentration. And so instead of trying to estimate from data with this dynamic model, we've transformed the problem into a static map identification, where for instance, if you focus on the rate number I, so the chemical reaction number I we want to model is rate, then you have to only perform this nonlinear least square optimization, for instance. And Ym is basically the kinetic model we want to fit here. Okay, so I'm not going now to talk about a dynamic system. I'm more going now to talk about static identification of this kind of system. Okay, so if you haven't understood a lot of things here, it's okay, there's no problem, because from now on, we are going to more focus on the modeling method on this. So now we, if you really didn't understand all the biological parts, it's okay, you can start following the presentation now, because basically the problem we want to solve is this one. If we focus only on one rate, we are going to simplify the presentation by focusing only on one rate. And so the identification problem we want to solve is this one, by computing the parameter eta star, which minimize the modeling error here. And where here Ym is basically this. So X is a multidimensional input, Y is our scalar output, and here you have the parameter mu j and theta j we want to identify. Okay. The problem is that if we want to start, if we do the non-linear least square optimization, we are going to get some local minimum issue. So we could consider some regularization method. But here, the idea that we have been following here in this project is to perform an intermediate modeling where we are going to model each modulation function here as with a Gaussian process model. And then we will use these Gaussian process estimates in order to find an initial value for this, for both parameters theta j and mu j for each modulation function. So that's the intermediate step. And now I'm going to focus on this part of this modeling in this presentation. So basically, now the problem is like this. We have a static map given by this, where Y is the output. What we want to model is a product of functions depending on the input, xg. And we are going to assume the additive noise assumption with a wide Gaussian noise of variance sigma e square, sorry. And each function hj will be modeled as a zero mean Gaussian process like this. And kj is the covariance function, is a covariance function that we have to choose. It can be parameterized by hyperparameters, but we have to do this choice in order to do the modeling. And we need a good choice of the covariance function, of course. And here, what kind of choice do we need is that, remember that hg can have 4 different values. If I go back here, these 4 functions. So we would like a covariance function which models very well these 4 types of functions. Okay. So in the literature, a kernel has been developed in order to try to model these 4 types of kinetics. So it's this expression here, and here you have 2 hyperparameters that I highlighted in red that you need to optimize before doing the GP regression, a Gaussian process regression. And here I give some, so sorry, it's actually a figure coming from this article where we show how that this kernel is kind of good in order to model the 4 types of kinetics, because here you have some random function which have been generated from this kernel with given values for both of these hyperparameters. And what you can see is that we sample here a double component. Here we see that we sample something that is close to an activation function, here something close to a neutral function, and here something close to an inhibition of function. So this kernel seems to be quite well appropriate for the modeling of the 4 types of kinetics. And in order to confirm this, so what has been done here is that we consider noisy data from an activation function, then we try to model it. And in blue, you have the posterior mean, and you see that indeed it seems that we have an activation function. Here we did the same with inhibition and double component. So it seems that this Gaussian process is very well adapted for this kind of model. But of course, when we want to try it on real life data, some problem may occur because the data you will obtain from the bioreactor have some bad properties. First, you can't have a lot of data because performing a measurement is really expensive. Moreover, you may be in a stage of your cells where the concentration do not vary a lot. So in terms of modeling, what does it mean is that we may have small data sets which are poorly distributed in the input space. And the problem is that if you want, then try to use the Gaussian process regression method with this covariance function of the literature. And you tried on, for instance, this very simple example where I have an activation function, 5 data, which are sampled from this small interval. Then here what you have is the posterior mean that has been computed from the data. And here you can see that indeed we have some problem in the modeling, but it's not really a problem of the covariance function. It's kind of a problem we will meet for every identification method, because with poorly distributed data, we can't really have very good estimates. So what can we do in this case? Well, the first idea would be to perform an experiment design, get more data points, but this is costly and this is very long to do. So the other method, the other solution we can consider is to try to incorporate the most prior we know on the model into the model, sorry. So that's what I'm going to propose to show you here. I'm going to show you a design of kernel of a covariance function for the GP regression, which will fit better to monad function by incorporating more prior. So it's kind of abstract what I'm saying here, but the idea is that we are going to incorporate the structure of monad functions in the design of a covariance function. And I'm going to show you how we are going to do that in the very simple example of an activation function. So let's consider this system. We have to estimate this system. We have to estimate theta. So here, of course, we can use directly nonlinear least square optimization. We can have a very good estimate of theta. But let's not do that. Let's propose a GP regression of this function. Okay, so we have to determine a covariance function. So let's calculate the covariance I will obtain between 2 output data obtained from 2 different input data at 2 different instants t prime. So the covariance will have this expression. And here we have the assumption that the noise is independent from the input. Okay, so now this would be our ideal kernel covariance function for the GP modeling. So this will be our ideal kernel. But why am I saying ideal and not the kernel that we are going to consider? Because this kernel is invalid. Because when you are going to consider different inputs, you will obtain a covariance matrix, which will always be degenerated. The rank of your covariance matrix will be always equal to 1. And then we kind of need this covariance matrix to be invertible in order to do the GP regression. So we have some problem here. The second problem is that it depends on the unknown parameter theta. So that's also another problem we have to solve. So the idea we have considered here in the mode for the design of the kernel is that we integrate the ideal kernel between two bonds, theta minus and theta plus. And so for activation functions, this is the kernel you obtain when you solve the integration here. So the nice thing is that then the kernel we obtain here is valid, meaning that if we have different data, if you have an input data set, you will construct a covariance matrix which will be invertible. So that's nice. However, by doing the integral here, what we did is that we introduced two hyperparameters, theta minus and theta plus. So we kind of increase the complexity of a GP regression. But here remember that theta is a biological parameter. And so how can we choose these bonds theta minus, theta plus? Well, here, what we consider in the project is that we consider bonds in which we have realistic parameter. In other words, we considered theta minus equal to 0.01 and theta plus equal to 100 because considering values of theta below 0.01 is not biologically realistic, and the same for parameter above 100. So if you don't have any prior, if you don't know anything about your parameter theta, you can choose theta minus equal to this and theta plus equal to this. So we don't have to optimize this hyperparameter, we just have to use these values. But, and this is where this kernel is quite interesting to use, because if you have some priors on theta in the form of uncertainties intervals, so for instance, theta should be between two bonds, let's say, I don't know, 5 and 10, then you should choose theta minus equal to 5 and theta plus equal to 10. And so this kernel is quite nice because we can include this kind of prior of the form of uncertainty intervals in the GP regression. So I'm going to show you how it performs in reality. So here we go back to the previous example I showed you. And here we did a GP regression with these 5 data distributed in this small space that you can delimited by these 2 vertical lines here. And what you can see in blue is the posterior mean computed with the new kernel. So what you can see is that it performs better. And it's kind of normal because in the kernel design, we have incorporated the structure of the function we want to estimate. So we included prior. So it's normal that we get a better estimate. Okay, now let's assume that we have, I don't know, we have contacted a professor in biology and it told us, okay, for this kinetic parameter, I know that it should be between 5 and 20. So then by choosing these hyperparameters like this, with these bonds of this uncertainty interval, you see that we can even get a better model. Okay, so that's how we can model activation function. If we want to do to design a kernel for inhibition function, we can do the same approach. So this is the expression we will obtain for inhibition function, except that now I also have to introduce 2 additional hyperparameters, mu plus, mu minus, which are the bonds where we do the integral. For the double component, remember that it's basically the product of the activation function and the inhibition function. So basically the kernel, we will consider the product of both kernels that we have designed before. And finally, for the neutral effect, so this one is kind of complicated because modeling with a Gaussian process a constant function is not really a good idea when you consider a covariance function which is not equal to 0. We should consider a mean function instead of a covariance function. What we did instead is that we approximated the neutral function by this with a very small epsilon, and then we construed a covariance function for the neutral effect like this. We could have considered a mean function, but here for the simplicity, for the sake of simplicity, we consider more covariance function by doing this approximation. Okay, so for the 4 types of kinetics, we have designed a better tailored kernel. So that's good. But the problem is that when we are going to do the Gaussian regression, the estimation of each module, we don't know what type of kinetic we will have. Is it an activation? Is it an inhibition or double component? We don't know. So the final kernel that we have proposed here is to consider the linear combination of the 4 kernels we have developed before. And the linear coefficient beta here are positive coefficients, and they are hyperparameters that we have to tune. And as an example, if we denote by beta g the vector containing the hyperparameter here, if I choose beta g equal to this, then we will consider more activation function. And of course, this parameter, this hyperparameter beta, we are going to estimate it from data. We don't know beforehand what value should be optimal. What we are going to do is to tune the hyperparameters by using the data we have, by using the method called empirical bias. Sorry. So the idea of empirical bias is we are going to find the hyperparameter vector which maximize the likelihood of the data we have. So since we have n different metabolites in our model, we have n modulation functions to estimate. So our system is the product of n functions. And since we have designed a kernel for each hg, which is basically this kernel, this sum here. So we have n beta g, hyperparameter vector beta g to optimize. So by denoting beta, the vector obtained by concatenating all parameter vector beta g, the bias empirical method is like this. We want to find beta such that we maximize the likelihood here. But the problem is that because of the modeling we have considered, here we have product of Gaussian processes. And so it is, the likelihood will be intractable. We don't have a closed form expression of the likelihood in this case. And so we can't really perform the optimization directly like this. So we have to find a way to circumvent the problem. And the idea that is often done in GP regression is to introduce latent variable in order to simplify the modeling. So latent variables are variables that are hidden in our model, and this variable here are indeed hidden in the model because we have the data of y, we have the data of x, but we don't measure h. So we are going to consider this as our latent variable. So these are basically the latent variable we are considering. So we have n data, and since we have a small n modulation function, the number of latent variable we are going to introduce is capital N times n. And now how we perform the hyperparameter optimization. So here I really simplify the explanation because I'm not going to enter in details, but it's basically an iteration, iterative estimation of the hyperparameter and the latent variable as like this. So you start your algorithm with an initial value for the hyperparameter vector. Then from this hyperparameter vector, you sample the latent variable from the posterior here. You obtain estimates. From these estimates, you estimate the parameter vector beta, which will maximize this quantity. So since this is a Gaussian process, since we assume Gaussian process, we know this expression here. The only thing that we have to be careful is how to sample this, because this is again intractable to sample directly like this. So in order to perform this and to perform also the sampling, So in order to do this, we are using the expectation maximization algorithm and the sampling technique. Why the expectation maximization algorithm? Because it has a nice property, but it's an heuristic property. We will always have a convergence to a global optimum, except in rare cases. So that's why here, in order to perform this, we have expectation maximization algorithm. Then, and this is where I have to talk about the intractability here, is that we have to sample from this distribution here. But the problem is that it's intractable if we want to find a closed form expression or use, for instance, the inversion method for the sampling. So here, what we decided to consider is the Gibbs sampling. The idea of the Gibbs sampling is like this, is that you are going to sample from the conditional distribution of this joint distribution here. The idea is like this. So you fixed all your latent variable except one, you sample this variable. Then from this estimate, you sample the second variable. Then you do that iteratively until you sample the last variable here. And then you restart again by sampling H1 from the previous estimate. And so you are going to do that iteratively. And what you will obtain at the end is a stationary distribution, which will be equal to the joint posterior from which we wanted to sample at the beginning. Okay, so that's how we solve the sampling. And then after doing the sampling, how to optimize. So this step here, the estimation of the hyperparameter here with the maximization. So basically, we just have to solve this maximization problem for each modulation function. And so it has, since we have considered Gaussian process on each modulation function, we have this well-known expression of the likelihood here. And just a note here is that the covariance matrix K that I estimated, that I have here, sorry, have this form. So it's basically the linear combination of the covariance matrix of the kernel that I have designed before. And here the parameters are here. So they are linear in K, but what you can see is that here we have an inversion and here we have log det. So it's a non-convex optimization. So how to solve this? Here we consider to just do the brute force optimization like this, or do an initialization by doing a combinatorial optimization. So what I mean is that we set all the hyperparameters equal to 0 except 1. Then we solve this and we have a closed form expression for this. Then we do the same for the inhibition and we set the other one equal to 0. Then we do the same for the double component. And finally, for the neutral function. And then what we do is that we use the different beta that we have obtained to initialize the brute force optimization. Okay, so now that was for the method. I'm going to give some, we are going to see how this method performs in some numerical example. So first we are going to look at a toy example that I generated. So we are going to consider a rate with 6 metabolites and a noise which has a variance of equal to this. The type of kinetic we have considered like this. So we have 2 activation, 2 inhibition, 1 double component, and 1 neutral effect. And here you have the true value of the parameters we have considered for the simulation of the data. And here you have the number of data we have generated. So here we considered 30 concentration data chosen randomly in this interval. So 30 data seems to be kind of a lot of data, and this interval seems to be kind of big. But here, remember that we are considering data in a 6-dimensional space. So at the end, this data set is not really great for the modeling. So that is why here we are going to consider a GP regression with the new kernel design. So these are the results for each modulation function. So in red, you have the true system. In black, the posterior mean of the kernel in the literature. And in blue, the new proposed kernel. So what you can see is that the new kernel performed very better, except for instance, the neutral function. Here, what you can see is that we kind of estimated the double component, what we should have more estimated the neutral. Here also for the modulation function, we have some very large error happening here. But nonetheless, we see that we have very good modeling performances. So that was a toy example. Then we tried this on real life data that we have obtained from doing experiments with small bioreactors in the lab we have here at KTH. So in order to interpret this graph here, I'm showing 6 rates. The x-axis are the time. And so you see big jump here is because we have we have 15 different bioreactors. So here it's one condition, here it's another bioreactor, so that's how you have big jump. So basically here in black, you have the data. In blue, it's the kernel, the posterior mean we obtain from the kernel in the literature. In red and magenta is the new kernel. The difference between red and magenta is that red is the model obtained by doing combinatorial optimization of the hyperparameter and then brute force optimization, while the pink, the magenta data cross you can see here is only by performing the brute force optimization of the hyperparameter. Okay, so I think it's time now for me to conclude this presentation and to give some possible extension of this study. So what we have done here is that basically we have designed a better tailored kernel for the modeling of monad functions in order to model better the rates of the chemical reaction in a bioreactor. We have obtained better results than kernel in the literature. However, the results I show you here are only first results. We have still a lot of things to analyze here, but the result we have here seems to be promising. What we need, the next step here in this project is to do then the parametric identification to see if we will have a better estimate. And the main idea here for the design, or why did it work very well, is because we have incorporated the structure of the functions we wanted to model in the Gaussian process regression. Basically, what we have done is that we put a lot of prior in the modeling. And so that's why it worked, even if we have data which are kind of challenging, not well rich in some sense. Okay, so the possible extension we can consider of this study, because here it's really specialized for the monad functions. But first, a possible generalization we can do is that, remember here I did an integral between 2 bonds. Instead of doing that, we can consider any prior distribution and do, like, for instance, I don't know, a Gaussian distribution on theta, and consider and generate new kernel like this. Of course, you need the integral needs to have a closed form expression. The second possible extension, and that maybe that will interest the modeling community, is that assume that we have a model static map which is like this, where phi j are parameterized nonlinear basis function for which we know the structure, but we don't know the parameter theta j and we want to estimate it. Then we could use this idea of constructing the kernel in order to do GP regression and to tailor better the kernel design here in this GP regression. Okay, thank you. I would like to thank you all for your attention.
Thank you very much, Kévin. It was a great presentation, very clear, and moreover applied to biological data, which is always interesting for our community. Thank you very much. Are there any questions from the audience? Well, I do have one. In meanwhile, then I will let Vincent ask his. You are comparing your own kernels with the kernels from Wang in the literature.
Yes.
I probably missed when you define those kernels by Wang.
So the kernel is the one I showed here at the beginning. So this
Oh,
expression.
it was this one.
Yeah.
Okay, thank you. Thank you. Vincent, you have a question?
Yeah, thanks a lot for the presentation, Kevin. Is this, it was a question that related when I looked also in tailoring kernels. Do you know if that has anything to do with the green functions? Or maybe you know about? Because
no, I don't know about it.
The expression look a little bit like that, and that's what I do. So the green functions are used for tailoring kernels which you want to have specific properties. So it's more in the math literature, if I remember. And to be fully honest, I really did not completely understand it, but somewhat the expression looked kind of similar to what you have. And the second thing is that also what I found really impressive is that you have a multiplication of function, and from what I understand, this is what causes your problem, because as long as it's a sum of function in terms of kernel, you don't have any problem with that. But when it's a multiplication of kernels, the problem is that the straightforward answer is you multiply kernels, but multiplying kernels does not give you a solution in terms of multiplications of function. Yeah. So if you take k1 times k2, it gives you any possible solution in terms of sums of k1 times k2 plus k1 plus k2 plus, well, whatever. Yeah. So it's really hard to constrain this. Did you search on constraining really this multiplicative form? Because it's nice to search for, well, searching for the proper kernel is one thing, but searching to restrict your identification to hand out what is an explicit multiplication of function, this is something I didn't see in the literature. I struggle a little bit with that, and I never really found a proper solution. So did I miss something in the literature, or do you have something interesting to propose in that sense?
I haven't really considered that issue. The problem that can arise here directly is a problem with the data. If you consider the prediction error identification framework, for instance, if you have a problem of data informativity, like you don't have enough richness in your data, you can have like a lot of problem in the modeling. Like that may be what happened with the black one here, is that if you do the multiplication at the end, and I looked at it, you have a very good model.
Yeah.
But the model itself, the latent variable, are kind of bad. So this could be explained by a richness of data. And I haven't studied really the effect of this new kernel with this multiplication and with this identifiability issue, but that could be really the next step also to verify that the result we have here is not like a lucky result because I don't know, the data were good at that moment. But yeah, this is really...
And basically, we could sum up the result that you show here is that the multiplication of black curves gives the same result than the multiplication of blue curves.
Yes.
But basically, since you gave bounds on this theta plus and theta minus and certain shapes of the kernels, then it restricts to a certain viability of your function at hand.
Exactly. And here, yes, there's something that I would like to consider. Is that I'm going to show you again the graph of the monad functions, but for instance here, where are they? Monad functions are functions which are between 0 and 1. So that's something we should include, and I'm currently looking at this because what you can see in the result I got here in this very simple toy example is that here I have data which are above 1, which is not what we want. So
but it seems that it copes with the true function. Then I don't. Oh, you mean okay at the side at the border?
The true function is you can't see it, but there's a 1 here, and it won't go above 1. But here, we maybe by constraining the amplitude between 0 and 1, we can have a better result. And also for the black one.
But it's already quite impressive, especially if you have only 10 points. I mean, I don't understand how it is really possible to from 10 points of data to estimate
No, no,
12
it's not 10,
parameters?
it's 30.
Oh,
Here in the axis is the concentration amplitude.
okay, yeah, but it's still very impressive. Yeah, yeah. Thanks a lot.
Thank
Okay,
you.
thank you. Thank you very much, Vincent. Thank you very much, Kevin. Thanks for the answers and the questions. I have just a few remarks before concluding. This morning was announced the next French automatic control workshop, which is organized by SAGIP and GDR Max. So it will take place from the 23rd to the 25th of May in Biarritz, and hopefully it will be on site. So we didn't discuss yet with Xavier and with Mathieu Polican, but we probably may be part of it, and we will be very happy to meet you all in, I mean, on site, en présentiel. Yeah. I wanted also to thank all the participants. We are still 12 right now. We went up to 41 participants during this afternoon. So I wanted to thank you all for staying with us until the end. And I wanted, on behalf of Xavier and Mathieu Polican, of course, to thank especially the presenters, Martine, Abir, Vincent, and Kevin. All the presentations were very, very nice. Xavier, do you want to add something?
No, thank you to all presenters. I agree with you. And well, Biarritz, I really don't think it's a good idea. It's too much out of the center. Why not in Paris or in Lyon or whatever? The sort of, it's quite impossible to go over there. So, certainly, but anyway, so, yeah.
But there is beach and nice weather, so.
Yeah,
Actually,
I guess that's the reason. But that's.
Actually, the place is very nice. Indeed, there's beach weather. It will be
for vacation.
Summer. It's also the opportunity to meet other people from other groups from automatic control. So anyway, we'll discuss. Maybe we'll ask you also, all of you, whether you're interested with or not. And we'll take a decision to either to participate to that workshop or to do it in Paris as we used to do.
So, but yeah, that's to be discussed. But yeah.
Sure. Again, thank you very much for participating. And Xavier, if it's okay, we'll end the